Hi everyone, welcome to week 2 out of 3 of our protein section. Our first topic today is going to be protein biosynthesis. So hopefully most of this will be a review for you. So our learning objectives for this section are to define and discuss the components of nucleic acids, define transcription and translation, discuss the 3 RNA classes, and explain the concept of epigenetics and mechanisms of action. Okay, so nucleic acids are the components of DNA and RNA, and these are formed from repeating units called nucleotides. So nucleotides are also sometimes referred to as bases. And nucleotides are composed of a nucleoside and a phosphate. So the nucleoside has a nitrogenous, is composed of first a nitrogenous core. And this nitrogenous core is either a purine or a pyrimidine. So the pyrimidines here are 6-membered nitrogen-containing atoms, or nitrogen-containing rings, while a purine is a 5-membered nitrogen-containing ring attached to a 6-membered nitrogen ring. So first we have, and then the nucleoside also consists of a pentose sugar, either ribose or deoxyribose. And then in addition, the nucleotide is also includes a phosphate group. So here we see our nitrogenous base. In this case, this is a pyrimidine. And then our pentose sugar, the ribose or deoxyribose, and then our trusty phosphate group. And DNA is double-stranded while RNA is single-stranded. So there's 5 different nucleotides that are contained in the structure of a nucleic acid. So these are noted here, the adenylic acid, guanylic acid, and so on, but we most commonly refer to them just as their nitrogenous base names. And then we typically use just the single letter abbreviation most commonly. So we know these as adenine, guanine, cytosine, uracil, and thymine. And you can see that here we have adenine and guanine are both purines that are contained in both DNA and RNA. Cytosine is a pyrimidine that's in both DNA and RNA. And just as a review, the pyrimidine that differs between RNA and DNA is the uracil that's in RNA or the thymine that's in DNA. Now, we do have complementary base pairing. That is, we have the pairing of one purine to one pyrimidine. So in DNA, these complementary base pairs are adenine to thymine and cytosine to guanine. And in the RNA, similar, we have the cytosine that's paired with the guanine. But instead of the thymine, we have the pairing of adenine to the pyrimidine thymine. Okay, so how are proteins synthesized? Well, we have 2 basic steps that lead to protein synthesis. So our first is transcription and then translation. So let's start with transcription. And this is the process by which the base sequence in a strand of DNA is used to specify the complementary sequence in the bases of an RNA chain. So this, so the overall process is transferring the genetic information from a DNA to an RNA. So RNA is synthesized by the enzyme RNA polymerase. And this is an enzyme that's responsible for copying the DNA sequence onto an RNA sequence during transcription. So this is a complex molecule that's composed of protein subunits, and RNA polymerase controls the process of transcription, during which the information that's stored in the DNA is copied onto a new mRNA. so messenger RNA, and we're going to go specifically into what that is. And we actually do have 3 types of RNA polymerases in eukaryotes, but we don't need to know the specific types for the purposes of this course. So here we have this RNA polymerase enzyme, okay, and it's going down the DNA double helix, and in so doing, the double helix is unwinding. So here you can see on this, here we have the DNA double helix. It's unwinding the DNA double helix, and then we have RNA that is being formed by complementary base pairing of onto the second strand of DNA. So this RNA is very similar in its base pairings to the DNA, except for instead of the T, we have a U. And then in this way, the RNA polymerase moves down the DNA, making this new RNA. So the RNA, we have 3 different types of RNA. So the first is our mRNA, or messenger RNA. And this is a genetic coding template used by the translational machinery to determine the amino acids that are going to be incorporated into a protein. So we're going to go into a lot more detail about this, but basically when we are transcribing the information from the DNA onto our mRNA, then the mRNA will use this information to build a protein. Now we also have tRNA, or transfer RNA, and this type of RNA forms covalent attachments to individual amino acids and recognizes the encoded sequence on mRNAs for insertion of the correct amino acid. So basically, the tRNA takes the individual amino acids that are in the cytosol or cytoplasm, and then brings it to the protein producing machinery, where mRNA is telling the machinery how to build this protein, and the tRNA is bringing the individual amino acids to build the protein. And then we have rRNA, or ribosomal RNA, which is assembled with ribosomal proteins to form ribosomes that engage the RNA and form a catalytic domain for tRNAs to enter with amino acids. And this catalyzes all of the functions of polypeptide synthesis. So basically, the mRNA comes to the ribosome, which is assembled with rRNA, and then the mRNA can go through the ribosome to dictate how the protein is built, and the tRNA brings the individual amino acids to this entire complex so that the protein can be built. Okay, so once we have production of our mRNA strand from transcription, then this mRNA strand is going to leave the nucleus through the nuclear membrane, and the DNA is going to stay inside the nucleus. So the availability of genes on the DNA strand for transcription can be impacted by gene arrangement, gene amplification, or gene deletion. And again, we have just kind of a similar graphic to what we saw before. So we have the RNA polymerase. We're unwinding here and building the mRNA. And after the mRNA is built, then the code rewinds. The polymerase is moving this way, and we get our mRNA strand. So in order for genes to be activated for transcription, the chromatin remodeling must occur. And just as a reminder, the chromatin is the DNA that's kind of all clumped up in the nucleolus. And for this chromatin remodeling to occur, it's facilitated by a couple of different processes. So the first process is the acetylation of the histones. So just as a kind of basic reminder from our biochemistry classes, histones are a family of proteins that are associated with DNA in the nucleus, and they help condense the DNA into chromatin. So the nuclear DNA does not appear in linear strands. It's really condensed and wrapped around these histones in the nucleus in order to fit inside the nucleus and take part in the formation of chromosomes. So there's lysine amino acids in these histones, and the acetylation of these lysine amino acid residues in the histone proteins removes the positive charges and thereby reduces the affinity between the histones and the DNA. So this makes the RNA polymerase and transcription factors easier to access the promoter region of the gene. So therefore, in most cases, when we acetylate the histone, it enhances transcription, while when we deacetylate the histone, it discourages or represses transcription. So by acetylating or deacetylating these histones that are associated with DNA in the nucleus, we can either encourage or repress gene transcription. So we also have whether the gene is activated for transcription also depends on another process called, let me back up a little bit and show that this graphic is what's demonstrating the acetylation of the histones. So it's showing that when it is acetylated, then we have kind of the go-ahead for transcription of the gene, whereas when it undergoes an enzymatic reaction and it's deacetylated, then we have repression of this transcription. And you don't really need to know these specific enzymes, but what you do need to, the take-home message is that these enzymes are impacted by phosphorylation, just as many of our enzymes are. Okay, so now to move on to the second mechanism, which is the methylation of the bases. So this entails adding a methyl group onto the DNA specifically. And this methylation occurs at a highly specific location on the DNA. And the location is C, that is located right next to a G and linked by a phosphate. And so this is called the CPG site. So we can see in this little figure here, we have our promoter region, and this starts our transcription. But when we methylate the DNA sequence in a very specific location, then this represses the transcription. So these are just a couple of the methods that can impact activation of the gene for transcription. Okay, so now let's move on to translation. So this is the process by which genetic information in an mRNA molecule specifies the amino acid sequence in a protein product. So the overall process is starting with the mRNA and producing a protein. So how this occurs is that the mRNA, once it leaves the nucleus, it then attaches itself to a ribosome. And this is considered kind of the protein-making machinery. So, and then the tRNA is going to collect amino acids from the cell fluid and carry them back to the mRNA ribosome complex. And the mRNA is going to dictate the sequence on which the tRNAs can attach the amino acids to form the protein strand. And the genetic code for the amino acid sequence of a protein mRNA is in these 3 base sequences. So we'll go into that in the next couple of slides, but basically the mRNA has a 3 base sequence that will dictate for each protein that's going to be added on to this polypeptide chain. So this graphic just shows what we just talked about. We have our mRNA that leaves the nucleus, attaches onto this ribosome, which is protein-making machinery. It has a code. So for 3 bases in a row indicates a specific protein. The tRNAs bring these amino acids, dictates a specific amino acid. The tRNA brings the specific amino acids to the mRNA, and then we have a growing chain of proteins. Okay, So gene segments that get both transcribed and translated into a protein are called exons. So when the mRNA is making the protein, as the amino acids line up in the correct sequence, the ribosome moves along the mRNA and an enzyme bonds one amino acid after another onto the growing protein strand. And I do have a little video to show that I think will help elucidate how this happens. So when all of the amino acids have been attached, the completed protein is released, and the tRNAs go back to the cytoplasm to get more amino acids so that they can build more proteins with different mRNAs in ribosomes. At that point, when the protein is completed, the mRNA that dictated how the sequence of the polypeptide would then be degraded. And this whole process occurs very, very quickly. So in a cell, 40 to 100 amino acids can be added to a growing protein strand in only 1 second. So this is an extremely speedy process. Let's just watch this video showing how this all works.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double-clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
Okay, so that's kind of a really interesting way of looking at how proteins are synthesized. And then this graphic is just an overview, although a less active overview than we just saw. So DNA in the nucleus serves as the template. RNA is processed and then leaves the nucleus. mRNA moves to the cytoplasm and then becomes associated with the ribosome. And then the tRNA with the anticodon, so that's kind of the 3-letter code that's going to match up to the RNA to show what amino acid will be docked on there, carries the amino acids to the mRNA. Then the anticodon in the tRNA and the codon in the mRNA go through complementary base pairing, and then the polypeptide chain is transferred from the polypeptide. Yes, the amino acids are added onto the polypeptide chain to form this kind of growing polypeptide chain, and then the tRNA goes back to get more amino acids to build a protein. Okay, and this is probably a review, but basically when the protein comes out of the ribosome, it's in its primary structure. So its primary structure is just the sequencing and strong covalent bonding of amino acids that occur as the protein is synthesized from the ribosome. So what order are all of these amino acids in is its primary structure. But then once the polypeptide chain starts to grow, then we kind of get interactions between the side chains of these amino acids or on this polypeptide chain. And we get some weaker bonding, such as hydrogen bonding, which helps the protein to develop a secondary structure or a more complicated structure that has some weaker bonds to change how everything is kind of lined up and interacts with each other. Then we get kind of more complicated with our protein, and it has a tertiary structure. And many of the proteins in our bodies exist in tertiary structures. So this includes our transport proteins, our enzymes, and our immunoproteins. And a protein gains a tertiary structure when there are interactions on the amino acid residues that are considerable linear distances from each other along the polypeptide chain. So in the secondary structure, these weak interactions are occurring kind of close by. So let's say, just in this case, maybe between the amino acids that are close to each other. But then when it starts to wind up a lot, then we get kind of interactions between amino acids that are farther from each other, and it gives us kind of this more complicated structure, which is a tertiary structure. So this could entail the clustering of hydrophobic amino acids, electrostatic attractions between oppositely charged amino acid residues, and covalent bonding. So we have kind of weaker interactions here, and then we have stronger interactions here. And then we get to our most complicated structure, which is our quaternary structure. And this involves more than one polypeptide chain, at least two. And we have the interaction of these polypeptide chains through hydrogen bonding and electrostatic attractions. And there's different special regulation of polypeptides, and how these polypeptides interact confers different function. So one of our quaternary proteins is hemoglobin. Okay, now I'm not really sure what exposure you've had to epigenetics, but I did want to introduce it here. So nutrition epigenetics is a growing field, and what epigenetics is it involves the genetic control of which genes are going to be transcribed and translated by factors other than the individual's DNA sequence. So nothing changes with the DNA sequence, but what is happening is, or the idea is that other environmental factors that are occurring during the time of these transcription and translation processes can either switch genes on or off to determine which proteins are going to be transcribed. And this happens through a few different mechanisms. So a couple of them we have already talked about, which is the DNA methylation and the histone modification. And then in addition, another mechanism that could control which genes are switched on and off would be RNA-associated silencing. So these could be non-coding RNAs or RNA interference. And basically, by changing which RNAs are going to produce proteins, then these could trigger DNA methylation or histone modification. So epigenetics is bigger than nutrition, but nutrition epigenetics specifically, the basic idea behind it is that adequate nutrition is essential to development during critical periods of life, and early suboptimal nutrition, and that could either be prenatally or in the growth and development phases, could have, so this early suboptimal nutrition could have health consequences decades later. So some of the research that's been done on nutrition epigenetics has revealed evidence that nutrition epigenetics could play a role in the development of cancer, in the way that we age, in obesity or type 2 diabetes, and could also impact inflammation and neurocognitive disorders. So this is really interesting because it basically says that what happens to us in our early stages of development can really impact us decades later, possibly regardless of what our lifestyle choices are. And maybe not regardless, but maybe we kind of are, after we undergo the suboptimal nutrition early in life, we're just kind of predisposed to get these, to have higher risk of these diseases. So another interesting thing about nutrition epigenetics is that there's also research into not only our own exposure during critical times of development, but those of our mother or our grandmother, or even further on. How do their exposure to suboptimal nutrition during critical stages impact their genes, so that down the line we still may be experiencing the consequences of that. So this is all developing field, and there's a lot of interesting evidence out there. We are going to talk about one article for this week, and so hopefully this will just kind of introduce you to this really interesting field. Okay, so to get back to where we were, so the genetic code of the mRNA is read in a sequential manner, starting at the 5' end of the mRNA and moving towards the 3' end. And this corresponds to the N terminus and the C terminus direction of the amino acid sequence within the proteins. Now, a codon is, as we said, composed of 3 base pairs, and that is what dictates what specific amino acid is going to be added onto the growing polypeptide chain. Now, since we have 4 different bases, this confers in 3 coded chunks, I guess I could say. This confers 64 possible combinations of the 4 nucleotides, but we only code for 20 amino acids. So some amino acids are encoded by more than one triplet codon, and these can be shown here. So serine, for instance, can be coded by any of these 3 letter codons, or 3 base codons. Okay, so that brings us to the end of this section, and here are your practice questions, which I will let you do on your own. Okay, so let's move on to our next topic of the day, which is the feed-fast cycle and protein metabolism in growth and development and injury and disease. And, we're kind of nearing the end of this course, and in this lecture, we're going to kind of start tying some of the interaction of the macronutrients together. So your learning objectives for this section are to explain the 4 different stages of the feed-fast cycle, understand the body's energy budget, identify changes in protein metabolism in growth and development, and during injury and disease. So in the feed-fast cycle, we have 4 different stages that we refer to. So first is the fed state. I just ate a meal. After that, we have our post-absorptive state, which is also called our early fasting state. So after the absorption of our nutrients from the meal. Then we move into our fasting state, which is these kind of long periods of time, maybe overnight or a little bit longer, where we go without food intake. And then beyond that, we have our starvation state. So our fed state, it lasts about 3 hours after meal ingestion, so during the digestion of our nutrients. And this is just a diagram of how we utilize nutrients in the fed state. So I'm going to focus on amino acids specifically here. So in our fed state, our amino acids that we just ingested go to our liver. We know that from last week. For our digestion and absorption. And the amino acids can be shuttled out to our peripheral tissues, so our CNS, they can go to our adipose tissue, or they can go to our muscles. Or if we have extra amino acids, these can be converted into pyruvate and stored as triglycerides in our adipose tissue. So this is in our fed state. And then just for your reference, we also have how glucose and triacylglycerides act in this same fed state. So basically, if we have excess, it's going to be stored or used. Then we move on to our post-absorptive state, also called our early fasting state. So this occurs from the end of our fed state at about 3 hours post-meal and goes to kind of, if you could consider, a very long night without food. So 12 to 16 hours following a meal. So at this point, the tissues can no longer derive energy from glucose that's ingested. And instead, it's got to start relying on other sources for fuel, including glycogenolysis and gluconeogenesis. So this is nothing new for us. We've already talked about this. But during this stage, the alanine glucose cycle that we have talked about in our carbohydrate section is going to be really important. So pyruvate that's generated in the muscles and other peripheral tissues can be transaminated to alanine, which is then returned to the liver. So here we have that here. So we have our pyruvate that's generated in the muscle and is transaminated by alanine. So this transamination requires an alpha amino acid here, okay, and that donates an amino group and generates an alpha keto acid. So we have our amino acid. It's going to donate the amino group to the pyruvate. In that way, the pyruvate is converted to alanine. The amino acid that donated its amino group is no longer an amino acid because it donated its amino group, and instead it's an alpha keto acid, or what we might think of as a carbon skeleton. So the alanine that's produced in the muscle, sorry, my mouse keeps going on. The alanine that's produced in the muscle then goes to the liver and is deaminated with alpha ketoglutarate. So we have our protein here, we have our alpha keto acid here, and then the alanine is in this in this stage, then donates the amino group to the alpha glutarate. So then the alpha ketoglutarate becomes the amino acid glutamate, and the alanine is then converted to pyruvate. So then the pyruvate can be converted back into glucose in the liver here, and then which can then return to the muscle for energy. So so within the liver, the alanine is converted back to pyruvate, and it can either go back to the muscle for energy, or it can be used as a gluconeogenic substrate. It can be used as a gluconeogenic substrate, so which can then go back to the muscle for energy, or it can be oxidized in the TCA cycle. So we have that here, generation of ATP. Okay, so then the amino nitrogen from the amino acid is then converted into urea in the urea cycle, and then that's excreted by the kidneys. So this is the glucose-alanine cycle, and this is really important in this post-absorptive early fasting state. So during this stage, the primary source of fuel, or amino acids or proteins that are utilized as fuel, are our branched chain amino acids that are mostly stored in our muscles. And then other amino acids are released in lesser quantities from the muscles, including methionine and others that are listed here. Okay, and then this is how our macronutrients are primarily utilized in the post-absorptive phase. So again, let's focus on the amino acids, on the proteins. So in this graphic, the broken lines indicate the major substrate flow during fasting, and the solid lines reflect the predominant substrate flow during starvation state. So here we can see that in the muscle, we have alanine, and then the alanine goes back to the liver and is used to make glucose via gluconeogenesis, and then that glucose can go back to the muscle. So that's mostly what's happening in this post-absorptive state. Okay, and then we move on to the fasting state. So the post-absorptive state evolves into the fasting state after about 2 days without food. And in this state, the amino acids from muscle proteins, muscle proteins are broken down to provide amino acids that serve as a chief substrate for gluconeogenesis during this stage. And so amino acids are the main substrate for gluconeogenesis in this stage. So we do still utilize the glycerol from lipolysis and the lactate from anaerobic glucose metabolism some, but we're mostly relying on amino acids from muscle protein breakdown. So of all of the amino acids, for gluconeogenesis, we can utilize any of the glucogenic amino acids. And that's all except for 2 of the amino acids. And the only amino acids that are exclusively ketogenic, so can't be used for gluconeogenesis, are leucine and lysine. So instead, these are converted to ketones, and our tissues can adapt to using these substrates. So during prolonged fasting, our glucose levels drop, which signals the release of glucagon and glucocorticosteroid hormone, and this leads to a shift to gluconeogenesis. Okay, so we have our fasting, but then if we continue to deprive our bodies of food for several weeks, then this is considered starvation. And now our bodies are, in their infinite wisdom, know that in different stages we need to utilize different substrates for energy. So once we've gone several weeks without food, our body says, okay, there is a limit to how much muscle we can break down for energy. We have got to conserve the proteins just so we can continue our basic biological functioning. so that we can produce antibodies, so that we can produce enzymes, so that we can produce hemoglobin. So our body's saying, okay, wait. We're using way too much protein. Obviously, we're not getting any food. Let's start to conserve our protein. So then we have this shift again. So in an effort to spare our body protein. So during our early fasting state, we had a lot of gluconeogenesis from our amino acids. But as we progress to our starvation state, we're going to shift from gluconeogenesis to lipolysis, and our fat stores are going to become our major supplier of energy. So the brain can't use fatty acids for energy because they can't cross the blood-brain barrier. However, when we have lipolysis, we do get the release of glycerol, and that can be used for gluconeogenesis, which can supply our brain with energy. So this ability to produce glucose from the glycerol backbone of fats during lipolysis assures a continued glucose supply of fuel to the brain. This is just shown here. So yes, our amino acids can be used for glucose, but then when we move towards starvation, then our fats are going to be used to produce glycerol for gluconeogenesis. Okay, so this is just kind of a review from our last section on lipids, but it's very pertinent to our conversation now. So as we talked about in our discussion on ketogenesis, fatty acid catabolism produces acetyl-CoA, and if we don't have the precursors, which is oxaloacetate, for the TCA cycle to continue, then acetyl-CoA continues to accumulate and be converted into ketone bodies. Now, as ketone bodies levels rise in the liver, these can be exported as fuel to our peripheral tissues for oxidation instead of glucose. So as long as our ketone body levels are high due to fatty acid oxidation, the need for glucose and gluconeogenesis is reduced, and therefore our protein is spared. So when we were talking about ketosis before, we said that really the production of ketone bodies is kind of a two-sided coin, because ketones do help us survive during these times when we're not getting glucose. Our body can adapt so that it can rely more heavily on ketone bodies, and that helps us to preserve our muscle protein so that it can be preserved for other functions in our body. So the survival time in starvation does depend on the fat stores before starvation. So whereas an average person may be able to survive a month, or maybe 2 months, 1 to 2 months in starvation, a very obese adult could theoretically endure a fast over a year, but extreme ketosis could lead to early mortality. Okay, and then we have this same graphic, but this is demonstrating how macronutrients are utilized during the fasting and starvation states. So here we have our amino acids that then being conserved, so they're not as much of a substrate for gluconeogenesis. Instead, we're relying more on the breakdown of lipolysis. So that's these solid lines are indicating that that's the kind of the primary substrate flow during starvation. And so the fatty acids are going through lipolysis and producing ketones that then are utilized in our tissues for energy. Okay, so this is kind of a slide that's, again, pertinent to this topic, but also is starting to bring together what we've learned in this course so far. So we know that it's really hormonal control that's dictating how our macronutrients are going to be utilized or synthesized or degraded. And we have the opposite impact of insulin and glucagon. So in our fed state, the insulin is going to predominate. So this, so having being in the fed state, we produce insulin, and so we get rising amino acids from the diet, and then we have increased glucose uptake in the muscles, increased glycogen synthesis, decreased glycogen breakdown and gluconeogenesis. We have increased triglyceride synthesis and increased amino acid uptake into the muscle and other tissues, and increased protein synthesis in the muscle and other tissues. However, when we enter into our post-absorptive state, and then which will transition into our fasting state and our starvation state, then we have the predominant hormone is glucagon, and this increases glycogen breakdown, decreases glycogen synthesis, increases gluconeogenesis, increases fatty acid breakdown and ketone body synthesis, and increases amino acid uptake in the liver for gluconeogenesis. Okay, so so let's kind of talk about the body's energy budget during these different stages in the feed-fast cycle. So let's consider our fed state. So we've just eaten a meal, and let's say it's a big meal. We just feasted. So basically, we've eaten in excess of our energy demands and in excess of our protein demands. So we need a certain amount of protein in order to replace the proteins in our bodies and synthesize new proteins for enzymes, antibodies, etc. But when we eat in excess of these needs, then we have surplus protein. Okay, and extra protein is not stored as protein in the body. So a person can't grow muscles by overeating protein. And this is kind of interesting when we consider some of our culture of bodybuilding or I guess just exercise in general, where people are eating these vast quantities of protein thinking that it will improve their muscle synthesis. But really, the body first replaces our normal daily protein losses, and then we can, increase our protein oxidation slightly. But then beyond that, any additional protein is deaminated, and the carbon skeletons are used to produce fatty acids. So really, eating all this excess protein is just going to contribute to increased adiposity. Okay, so now let's discuss the biochemical metabolism during times of no food intake. So between meals, tissues such as the brain and liver just rely mostly on glucose for energy. And the fat stored in adipose tissue provides free fatty acids to the liver and muscles to be broken down for energy. And then also between meals, the body's carbohydrate stores, so their glycogen and their fat stores, have a sparing effect on the body's protein and amino acids. So they don't have to be used for energy, so that proteins don't have to be used for energy just during this time between meals. We've still got enough nutrients present in the form of carbohydrates and fats that we don't need to use our amino acids for energy. However, when we move into our overnight fast, then some of the amino acids in the muscles are converted to glucose to be used for energy, especially in the brain. So the longer we go without food, the longer we go without an input of glucose, we need to start relying on the amino acids in our muscles for gluconeogenesis. But, if it's just an overnight fast, this is completely pathologically normal, and we don't have really any negative effect on muscle mass or function in just the overnight fast. Okay, so then we move into our fasting phase. And this could occur during times of severe weight loss. It could be either intentional or unintentional. So it could be due to severe dieting or eating disorders, or it could be due to illness or injury, or it could be due to just lack of food in certain environments. So during this stage, we haven't had an input of glucose for so long that the body has no choice but to rely on muscle proteins to provide the amino acids that can be broken down for energy. So this explains why individuals with severe weight loss have lost a lot of their muscle mass too. So they may look like skin and bones, where you can literally see their bones because their muscle has been utilized for amino acids for energy because the body didn't have enough input of fat or glucose. So when we break down fat, or when we break down carbohydrates and we produce carbon dioxide and water, it's really no difficulty to the body, okay? But so that's when we break down lipids and when we break down carbohydrates. That's no problem for the body. We are well equipped to deal with those, excretion of those. So obviously water isn't a problem, and we have an easy method to expel our carbon dioxide. However, when we're starting to rely predominantly on our amino acids for energy, we kind of have a different situation, because when we break down amino acids, we have to eliminate the nitrogen from the protein. And this requires an additional step of urea production so that we can excrete our nitrogen in our urine to avoid the buildup of nitrogen in our bodies. Okay, so growth is an anabolic process that occurs throughout, starting when you're a baby and on to your adulthood. And this growth process includes a net increase in body protein, as we would expect. And this entails significant remodeling. So not only protein, so basically in order to have this kind of net increase in protein, we also have the degradation of protein. So we're kind of breaking down, building up more, breaking down some, building it up more, so that we have this growth over time. And so in newborn infants, our rate of protein synthesis is over twice that of adults. And this declines progressively throughout life. So it's the highest when we're infants, and then decreases as we move towards adulthood. Okay, so when children do undergo malnutrition, when they're recovering from their malnutrition, their growth rate is much greater compared to healthy children. So we have what's called catch-up growth. So the body knows, okay, I've been deprived of energy long enough, I got to catch this kid up. So they're going to synthesize protein at a much faster rate. And the rate of growth in the child is positively correlated with the protein synthesis, which makes complete sense. Now, increased protein synthesis in growth is, during growth, periods of growth, is accomplished with increased levels of RNA in the tissue. So the more our mRNA and our RNA we make, the higher capacity we have for making proteins. Again, makes sense. So at puberty and during pregnancy and lactation, we have appropriate hormonal control that changes so that we can have increased protein synthesis. So one example is that we know androgen is anabolic, which provides increased protein mass, and this is mediated in part through potentiation of growth hormones. So during times of growth, we're going to have more androgen, which is going to dictate more protein synthesis. Now, during periods of malnutrition, there are reductions in insulin levels and increase in glucagon levels, just as we would expect. We're not getting enough food, so our body is saying, okay, I need more, I need more. So glucagon is going to predominate. In this period, growth hormone levels are elevated and thyroid hormone levels are reduced, and there's conservation of energy and amino acids as seen in starvation. So malnutrition, even if we're getting maybe some food intake in, our body is still treating it as if we're starving, even though we are getting some in. So it's going to try to conserve our energy by decreasing our metabolic rate and trying to preserve our protein, our body proteins. So during this time, oxidation of amino acids is reduced, and protein turnover is reduced. So we're just trying to hang on to the protein that we already have in our body during times of malnutrition. So in children, malnutrition is accompanied by growth cessation. So this includes stunting, which is a reduction of height for age, and wasting, which is a reduction of waist for height. Also during periods of malnutrition, albumin concentration is reduced. So basically our body is saying, I'm not even going to produce as much albumin at this point because my amino acids are so precious that I need to hang on to them for other proteins. So albumin concentration in the past has been used for, sorry, I shouldn't say that. I should say historically, albumin concentration has been used to assess nutritional status, and it still is in certain settings, but I think the reliance on albumin to in order for us to diagnose malnutrition is kind of decreasing, because we see that in times of stress, such as injury and disease, which is usually, what's going to bring a person to the hospital, the albumin concentration is reduced anyway. So we can't really just attribute low albumin to malnutrition. So it's not as useful of a marker. Also, the half-life is pretty long, so we don't see a lot of changes during a short hospital stay. Okay, so one thing I just wanted to bring up in this lecture was the physical exam for malnutrition that is growing in popularity in the dietetics field. So it's kind of interesting to see, traditionally dietitians have been, strictly counseling, we're talking to each other, but we're moving really into a phase where in some fields of dietetics, we're actually doing physical exams. And one of these that is particularly important is the physical exam for malnutrition. So this chart just kind of demonstrates the specific areas that you examine on a patient during their physical assessment and how they would look in different stages of malnutrition. So when we look at, so I'm just going to go to the next slide here because I have some pictures that will hopefully help demonstrate this a little bit better. Okay. So here we have the examination of the orbital region surrounding the eye, and in severe malnutrition, it kind of has a hollowed look or like a sunken look with dark circles and loose skin. So you can kind of see that demonstrated here, this kind of hollow, depressed eye with dark circles. Okay, next we would look at the upper arm and triceps. So this would be like a skin fold we could do, or we could do that just with our fingers. And this will show us, I don't have a picture of this here, but you could actually see it with this woman here with her triceps, that if I tried to squeeze behind her tricep, all I would get was skin. It would indicate that there was no muscle there, and that's what's going to indicate malnutrition. We also can look at the thoracic and lumbar regions. So our ribs, our lower back, and basically in severe malnutrition, we would have the depression between the ribs. So that can be demonstrated here. And the iliac crest would be very prominent. So that's of the hips. So if we can actually see between these depressions in our ribs, both in the front and in the back, this would indicate malnutrition. We can also look for muscle loss. So we would look at that in, okay, so all of those that I talked about would indicate subcutaneous fat loss. Now, when we're going to just try to screen the patient for muscle loss, we look in a couple different places. So we can look in the temple region here, and this isn't the best picture, but in a malnourished individual, we would have the hollowing or scooping or depression of the temple, and you can kind of see that here, that we have kind of this depression on the temple. And if you feel your own and you're well nourished, that it would just be flat. But here, and we see this a lot in older individuals, or maybe in patients who have cancer, we have this kind of scoop looping or hollowing out of this in this temporal region. Okay, we also can look at the clavicle and the shoulder, the deltoid, trapezius muscles. So in someone who's malnourished, that would be really prominent. So you can kind of see that here. This kind of squared off shoulder is indicative. This prominent deltoid, these prominent clavicles. So in women, clavicles might be prominent anyway, just because of how you know, a certain body type is. So in a woman, you know, maybe that would be just kind of a piece of the puzzle when you're looking at the clavicle. And then our last one is the this portion of the hand where if someone is malnourished, we would kind of, you know, in a well-nourished person, this area, this has always been a hard one for me, but if you lay the hand flat in a well-nourished person, it would be in this area kind of, flat or even protruding, but in a person who is malnourished, we would kind of have more of a hollowed out region showing that there's been muscle depletion. Okay, so we just talked about malnutrition, so that's going long periods without any food intake. Now we have a slightly different situation during injury and disease. Okay, so loss of protein accompanies many disease states such as trauma, cancer, or infections. And our body treats our protein in very different ways than it did during malnutrition or starvation, in that our adaptation to preserve lean body mass during these disease states does not occur. So if we're malnourished or we're starving, our body says, okay, I got to preserve the protein that I have left. But when we have protein loss in these disease states, we don't have that protein sparing effect. So here we have kind of a, so the body protein continues to serve as a substrate for energy during these times. So this table, you can see kind of the comparison of how protein is utilized in these different physiologic states. So in malnutrition and starvation, we preserve lean body mass, but in injury and disease, we do not have this adaptation. In malnutrition and starvation, our energy is conserved by lowering our metabolic rate, but in injury and disease, we actually may be hyper-metabolic as our body tries to heal. In malnutrition and starvation, the body relies on ketones for energy in order to spare our protein, but in injury and disease, we do not have this adaptation, and therefore our body protein is not preserved. So during injury and disease, amino acids are available for gluconeogenesis and synthesis of liver proteins, and this is especially our acute phase proteins like our C-reactive protein and fibrinogen, which rise after injury and an illness. Okay, so I have just one final slide here that demonstrates everything that we've been talking about. So here we have inadequate protein or energy intake, okay? And our body adapts by reducing at first we reduce our protein stores and we have reduced metabolic rate. But then as we adapt, we then rely less on our body proteins, and we kind of reach this homeostasis where we can survive for longer periods of time. However, if we have metabolic stress or just very extreme starvation, then our body does fail to adapt and we continue using our body protein and our fat loss. We have lower rates of albumin or lower levels of albumin, and because we are not preserving our protein for other functions, we have immune deficiency. And here are your questions for this second section, and let me know please if you have any questions. I hope everybody has a great week.